you very much for the introduction. Again, my name is Jack Egelund Madsen. I'm the CEO of Diagonal Bio, a company that has come a really long way in a very short time. The company was founded in 2020. We brought in around 10 million from private investors. Last year, we already listed at Nasdaq First North Growth Market in Stockholm, where we brought in additional 40 million. And already this year, we are planning to initiate our commercial activities. So it's been quite an amazing journey, and of course we look forward to the future to come. Let me start by showing this picture. If I've asked you 5 years ago what this was, nobody would be able to answer this question. Today, I'm sure it has a lot of negative feelings when you look at this. And of course, this is defining the health crisis of our time, COVID-19. According to WHO, more than 500 million people have been confirmed infected with COVID, and more than 6 million have died with COVID. However, this is not the only virus or infections that can affect humans. Here I just listed some of them. Everything from Ebola, HIV, influenza, and so on. And the problem with this is that this is not limited here. There's a constant evaluation here, or evolution. New bugs are going to come up that can affect humans. And actually, there's a quote from this high-impact journal, Nature, and I will read it here. It states that every single time we have an outbreak, we are one single step behind in that we do not have rapid diagnostics to detect the new organism. I think this has been pretty evident during the COVID-19 crisis. The world needs high-quality diagnostics. And this is actually why I'm standing here today, to tell you about Diagonal Bio's first product. We call it Panviral. It has the potential to be the most accurate, easy to use, fast, universal diagnostic platform for testing any type of infectious disease. Let me show you. First of all, Panviral detects genetic material in real time with very high accuracy and sensitivity. Essentially, we are bringing the gold standard of diagnostics to point of care on your patient testing. We can do that without having the need for these external laboratories. As you can see here, it's a neat little box, basically half the size of a laptop. It's very easy to use and customize. It has a, you can say, a guided user interface to make it very easy for the user to actually maneuver. And I have a small movie showing actually how this works. First of all, you have this small box. It has a very easy to use touchscreen. We have 8 channels, and that actually means we can scan for 8 different diseases at the same time, or 8 patients. In each of these channels, we have specific primers who will identify specific regions within a disease. If they find it, they will amplify it exponentially. Then we have a sensor dipped into this solution, and we can detect that very precisely and very early on. And again, we can detect 8 samples in parallel. So why is this important? If you look into today's solutions, then most of these diagnostic solutions, they are diagnosed specific. I'm pretty sure you have all tried this with COVID. If you have flu symptoms, you go to the test center, and then you get an answer. COVID, yes, no. With Panviral, we can have a more holistic perspective. So the whole idea is here, because we have these 8 channels and we can scan for 8 different diseases at the same time, we will be able to make a respiratory panel. So I listed an example here. So when you go to the fall and you start to have these flu symptoms again, instead of getting this yes-no answer, within 10 to 15 minutes, we will be able to tell you potentially which type of COVID you have, if it's influenza A or B, or it's respiratory bacteria. We can do the same thing with, for example, sexually transmitted diseases. Instead of doing individual testing, we can test a whole battery of different diseases. This has huge benefits. This is just an example of how accurate we can test. This was a study we did before Christmas on COVID samples. And as you can see here, we have positive COVID samples and negative COVID samples. We detect this very quickly and very precisely. And as you can also see on this graph, we detected COVID samples below 10 minutes. Panviral enables detection of multiple diseases in parallel, or multiple patients, in less than 10 minutes. A little bit about the technology behind Panviral, because you could ask, so why didn't somebody else come up with this idea? What we are using is a combination of loop-mediated isomal amplification. I know it's kind of complicated, we just call it LAMP. With this technique, we can identify specific regions within a pathogen or a disease, and then we exponentially amplify that. By coupling it to electrochemical sensors, we can detect that very early on and very precisely. And of course, this is patent-pending technology with a priority date in March 2020. So nobody can copy this technology. This makes Panviral unique and unlike any other system on the market today, because we can combine very short time to positive, so below 10 minutes, as I just showed you. We have accuracy in line with gold standard PCR testing today. And on top of that, we have this holistic perspective. So we can test multiple samples within 10 to 15 minutes for a single patient. This is, of course, creating commercial opportunities at a global scale. So what are the problems with today's testing? As I discussed before, I'm sure all of you have tried to do this test for COVID. The PCR tests. They're very accurate, yes. That's the gold standard of diagnostics today. But they're also slow, complicated, and expensive. So what are the alternatives? The alternative is to use the quick tests. And I'm sure all of you have also tried this. They're very easy to use, they're cost efficient, but unfortunately they're not very reliable. There have been several studies who have shown this. There was a study last year from Statens Serum Institute in Denmark, where they compared more than 4,000 positive COVID samples identified by PCR, and then they did the same sampling with the quick test. Less than 2,000 was tested positive. FDA have also raised concern about these quick tests. Either way, if you have whether you have a positive or negative quick test, the recommendation was always that you needed to take a PCR test anyway. If you look more general on the diagnostic testing today, then most of these tests are outsourced to external laboratories. That means long response times. And again, I think all of us have tried this. Sitting and waiting on a result, whether you're positive or negative, creates anxiety. This could also be young people, I mean, like this picture here, waiting for the test from a sexually transmitted disease test. It also means delay in treatment. In worst case, that means that the patient can die or have unnecessary complications or prolonged recovery time. And of course, if you have false diagnosis, or it takes a long time before you get the results, there's a chance that you can spread the infections. I have a business background, but I also have a technical background with a PhD in biotechnology, and have been working at Copenhagen University within gene discovery as an associate professor. And I found a study that was done between Howard and Ohio University, who really illustrates this perfectly. This is just a short, you can say, overview of what they were doing, and I will try to make this even more simple. The study here was published in 2021, and it's based on COVID. What you can see on the bottom bar here is that if you do no testing at all in this study, then you have 100% spread of the disease. Then you can start to do frequent testing. In this case, if you do testing every 3 days, but it takes up to 2 hours to get the result, you will still have up to 45% spread of the disease into the group. If you, however, can do the testing in real time, and that actually means that you get the results immediately when you are at the test center, then you can limit the spread of infections to almost zero. And that's exactly what we can offer with Panviral. Panviral offers testing in real time within these 10 to 15 minutes. This means that Panviral can help speed up the diagnostic process directly near the patient without compromising the accuracy and sensitivity. This means quick and accurate diagnosis, optimized and fast treatment, and reduced spread of infections. And of course, this is a huge market. If you look into the diagnostic testing market today, it's valued at around $34 billion US. And I'm pretty sure on top of all of these, you can say this health crisis with COVID, this number is going to rise up. And it's also pretty sure that the COVID is not the last pandemic we will see. There will be much more to follow. And we need to be prepared. Currently, the diagnostic testing market is dominated by the quick test. But as already explained, there's a limitation here in the accuracy. Then we have you can say the gold standard of diagnostic testing, which is the PCR-based technologies. They're very accurate, but they're also slow, complicated, and costly. And then, of course, there's multiple other technologies out there. I stated CRISPR-based technologies here because it's a very capable technology, but it's also highly complex, time-consuming, and expensive. So where do we see the best niches fitting for Panviral? And here I want to discuss different options. The first one here is to make tailored regional specific panels. I already touched upon this in the start. So when we go to the fall again, we would like to make a respiratory panel, which means that if you have flu symptoms, you can go to the test center and then immediately get an overview of which type of COVID, influenza A or B, or respiratory bacteria do I have. It could also be that you want to travel somewhere exotic. And then you can make an exotic panel for when you get back home for this travel. And of course, there's also this where we could test for sexually transmitted diseases. Instead of having young people wait for days or weeks for the result, they could get the results within 10 to 15 minutes. The second I want to discuss with you is using Panviral to monitor infectious disease outbreaks. Panviral has very high sensitivity. And that means even before you would see an outbreak, you would be able to test that with Panviral. You can imagine governments using Panviral as a surveillance system. Or even that you have Panviral standing in the airports. So before you board a plane to the US, you can get tested. Or at big sporting events or concerts. The last niche here is about using Panviral for disease and treatment monitoring at the hospitals. Because of the real-time capabilities of Panviral, we would be able to validate treatment directly near the patient, which of course will have huge value for both the medical doctors but also the patient. So how do we then plan to sell Panviral? First of all, we have this nice neat box here. So this is Panviral. It's a universal system. We can use it to test any kind of infectious disease. The only thing we require is the presence of genetic material. So we can test Ebola, COVID, influenza, sexually transmitted diseases, or whatever. But to make this work, you need two types of consumables with the system. You need to have this, this panel with 8 channels inside, and there's also a sensor in that. It's a one-time use only. And with that, you then also need to have a buffer, which is specific for individual diseases. Those two will be the driver of the revenue. Because Panviral is a universal system, we can go into different disease segments. And that also means we have the option to go into differentiated pricing strategies. So how do we then plan to get Panviral out to market as fast as possible? And here there's multiple routes we will follow. The first one that I will describe for you here is the one that is listed here, and that's to manufacture Panviral as a research use only device. It has several benefits. First of all, it will enable us to go into the US market under what's called an EUA, emergency use application or approval. The reason that Panviral has this potential is that we can test some of the most deadly diseases in the world. This could be Ebola, for example. We could also test for antibiotic resistance genes or COVID. This is giving us a possibility to go into the US market even before we have the full regulatory package finalized. We will also look into other options like the Middle East or Far East, again, where the regulatory demands are somewhat slower or lower than what we see in the European market. But of course, at the same time, we will continue to build our technical documentation to ensure we get the CE marking according to the IVDR regulations. But I want to stress out that right now, everything we do inside the company, this getting the first initial manufacturing of Panviral done as a research use only, has top priority. This will enable us to initiate our commercial activities. It will enable us to start partnering collaborations with key opinion leaders, infectious disease clinics. It will enable us to start to generate scientific data and publication to support some of these claims. And that can be used in the technical documentation when we go for the full CE marking according to the IVDR regulations. We recently published, updated milestones, just to make it very clear what it is that we're doing inside the company. I don't want to go through all of them today because you can find them online at our website. But I want to stress out again, our primary focus in the company right now is to get the initial manufacturing of Panviral done as soon as possible. We are, of course, also looking into other activities like studies with clinical samples and so forth. All of this is, of course, only possible because we have a very strong team. So beside me in the company, we have Per Henter as the Chief Financial Officer. He has a strong background and have been working for other listed companies before. Then we have Andreas Nyberg, who is one of the co-founders and have strong knowledge about the science behind all of this. And of course, we have a very strong and a dedicated operational team who's pushing hard every day to ensure we get Panviral to market as soon as possible. And of course, all of this is supported by a very professional and dedicated board. So with that, I want to say that our vision is to limit the spread of infectious disease worldwide by creating an accurate, easy to use, quick, cost-efficient, universal diagnostic platform for testing of any type of infectious disease. It has been a quite remarkable journey so far. And of course, you're welcome to follow us and also follow us on this next year, couple of years, where we will get Panviral to market. It's going to be really exciting. And then I think I will be ready to take some questions. Thank you.
Jack, thank you. Thank you. Fascinating, fascinating.
Thank you.
The big question is, and this might come from, have you seen the Theranos Apple TV Plus series about Elizabeth Holmes?
Oh, I'm sorry. I didn't see that.
Yeah, Theranos, the blood test company. Yeah. She's facing her sentencing now. She might go to jail.
Oh, the one in the US. Okay. Yeah. I saw that company.
They had the box, but they ran the tests on Siemens machines, and it actually never worked. And George Schultz's nephew was there, and the Wall Street Journal broke it. And
I've heard that story before. And
I think they came to $10 billion valuation, something like that. But then, of course, plunged to zero. So. No other reference to you, but it seems that your machine is a bit digital. I know you have a prototype. I will let you answer in length on this.
Okay.
Your machine is a prototype. You have several prototypes. But if the final machine reaches antigen accuracy, you are actually not very far along. Then you don't contribute so much, and your success will be limited. I guess not zero, but it will be limited. But if you reach what you say, PCR, 10 minutes, multiple testing, I would guess this is huge. But you had I couldn't quite make out from your slide of key events.
You want me to?
Yeah, if you want to,
Yeah.
please elaborate on where are you with the prototype, how far along will you go, and how certain are you on how far your machine will reach in terms of speed and accuracy?
There was many questions in one
Yeah.
question, I think. And first of all, I don't hope we will not be compared to Theranos. And that's also why we put it as one of the key objectives in the company right now, is that we want to do everything we can to get Panviral to market. Because that's going to take away all the doubt. That's for sure like that. So that's the thing.
I agree.
And of course, we will do everything we can to do this as fast as possible. We are planning to come up with a more exact date later on, so I will not come with that date today. But the plan is to initiate our commercial activities this year. That much I can say. And I will also say that if you look from the time that we listed the company in July 2021, we have reached all our milestones so far. So we are on track, and we have done everything we promised to the market as well. In terms of the accuracy, you can say, but that's the whole idea behind Panviral. And we are fully aware of that. And we are, of course, do testing in-house all the time on our prototypes. And we are seeing very good accuracy. I mean, we know that that's the whole, you can say, business case with Panviral. That is that we have the accuracy and we have the speed. And
yeah, so is that a correct assumption for us, that if you don't hit the accuracy, your success will be limited, but if you hit it, it's a big thing?
We have already done studies as well, and also that has been published, that is available online if you check on the website, talking about the accuracy. And we of course keep optimizing that. So we have that, and that's, you can say, that is the whole business case. Speed, accuracy, and then we have this where we get a holistic picture of a disease area. Instead of getting these diagnosis-specific answers, yes, no questions, we can answer a whole range of different diseases within one area.
Yeah.
So that is the whole business case, yes.
Okay. If we pull up the slide with the multiple positive results, you have it's many lines on the slide, and you have negative and positive.
Go back. This one?
No, the dotted lines. Sorry, you have a slide with line charts. There are positive results, but there are many results.
In the very beginning, I believe.
There.
Ah, this one.
There.
Okay. Yes.
Elaborate on this a bit.
Okay, so this is the way that we are measuring. So we are measuring current in the samples. I would say that. And what you can see is in a negative sample, because we don't have amplification. So actually, maybe I should go to this. Go one forward. Yeah. The way that Panviral works is that, let's say we want to scan you for COVID. Then we have very specific pieces of DNA that are looking for a specific marker within the COVID genetic material. If those primers find that target, it will be exponentially amplified. And that creates a current we can measure in the sample. And the way that we measure it is, of course, using electrochemical sensors. But if you don't have that so if you're not infected with COVID, the primers will not find the target, and therefore there will be no amplification. And that's also why, if you go back, you can see that there's no, the curve is not rising in the negative samples.
Yeah, and this arrow here, you call the positive.
Yes, we make a threshold.
Here, it's distinguishedly, it's big difference between negative.
Yes.
Yeah, and then this happens.
Yeah.
Okay. Go forward one.
Yeah.
Oh. One more. Is this no?
Yeah, sorry.
I'm sorry, Jack.
No, that's okay.
This is the patent pending, the electrochemical analysis.
The patent is about how we combine the two technologies. This is a molecular biology method, and this is electrochemical sensing. When you combine the two, that's what the patent is all about. Okay. And because we're using electrochemical sensors, we can detect this amplification extremely fast and accurate.
But in order to market this, we need a CE mark, don't we? And that takes some time.
That's what I tried to explain before. So we are moving on multiple tracks. So if you want to go into the European market, we will need to follow the new IVDR regulations. And there we will need the CE marking. That's absolutely true. But because Panviral can test some of the most deadly diseases in the world, the path to market is, you can say, somewhat easier because we can save lives. So FDA has a list. I think there's 10 or 11 viruses or bacteria on that list. And if you can test those, you are allowed to, you can say, get easier access. You don't need to do the full CE marking. So that's why I had this the slide with the regulatory path. We will move on multiple different paths. We will get the research use only out as quick as possible. So we get somebody else to endorse the system, show scientific data coming out of universities or infectious disease clinics. That can also be used if you want to market the product under this, what's called an EUA under FDA, because we can test some of these deadly diseases. At the same time, you can still market the product in the US, or sorry, in the Middle East or Far East, where the regulatory burden is somewhat lower than what you would have in Europe. And of course, all of this data we will generate while we have this, we can use that for the application that's needed when we do the, you can say, the filing for the IVDR regulation under EU. So this is not only one. We have multiple tracks to run on, and we can go to market with the research use only as well. That's the plan.
Another more complicated thing is, I mean, if we're looking COVID-19 or not COVID-19, that's fairly simple. But if I take a sample from a urinary tract infection, I have 8 tubes, and I want to know, is there a bacterial infection? Then I need to know what specific bacterium I'm looking for, because I can only put one sensor for a specific bacterium in each of those 8
and that's
bottles.
absolutely true. But today, otherwise, if you want to find out the bacteria, typically what you want to do is you take it on an agar plate, and then you have to wait 3 days to the result. Now, with this technique, if there's a bacteria and you know what it is that you're looking for, which typically you have an idea, we can give you the answer within these 10 to 15 minutes instead of waiting days for bacteria to grow up on a specific medium.
Yeah. Short one.
Yeah. But if I'm a general practitioner, I would like to have that machine. I would like to know, is it a urinary tract urinvägs infection?
Yes.
Yes or no? I would like to know what bacterium it is, and I would like the answer in 15 minutes. Can I get it?
You would need to know what you're looking for to get the answer. So the machine, again, when we design these primers, we need to know the genetic code. So we need to know what it is that we're looking for to get the answer. It's not a universal machine in that sense that you just put in a sample and then you get all kinds of answers. It can only give you the answers if you know what you're looking for.
Is this present or is it not present? That's the answer you give.
Yes, yeah, that's
Yeah.
yeah.
Okay, any final question to Jack from Bio? I think we're running behind here, even though we have plenty of time. Jack, fantastic presentation.
Thank you.
Thank you very much. Thank you, Jack.
Thank you.
Jack from Bio.